AV nodal cells and convoluted tissue, but most fundamentally I'm going to talk about the cardiac myocytes. Calcium. I presume Professor Kovács already discussed the anatomical topic of the ventricular myocyte, the cardiac myocyte. But apart from this function, how the calcium governs the contractility of the ventricular cells, calcium has many different functions in the living cells. Referring to the first chairman of the Department of Physiology 70 years ago, he said, if you are a researcher and you are happy life, don't work with calcium. Because calcium does everything and the opposite too. It exerts such a complex regulation of the living cells. 70 years ago, the complexity seemed just simply unhandleable for the researchers. Today, we accumulate the large amount of knowledge of the calcium function. We still see it's too complicated to understand. Probably in the next 50-100 years, it will give a lot of job for the researchers. It regulates contractility, energy consumption, apoptosis, and the law. And in such a cell like the ventricular myocyte, which is practically contracting machinery only, majority of the cell's protein are contractile proteins. This regulation, as you will see, will regulate most of the contraction, but apart from this, a lot of other functions too. This is the stage for the eons. Part of it, the calcium cycling, was discussed with relationship to the easy coupling. Today I'm going to spread a little bit the focus, and we are going to other functions, not on the contractility, but we are going to focus on the contractility mainly. Homeostasis. What is homeostasis? According to the definition of the early physiologist Hammond, homeostasis is a property of a system to maintain balance. And this is what we want to emphasize. In the earlier lectures, we already discussed several components of the calcium homeostasis, channels, pumps, and other functions. Today, I would like to emphasize the homeostasis, how the balance is maintained. When we approach this topic, when we want to understand how the calcium exerts its regulatory function, instead of believing that we have a fine-tuned system where the amount of calcium determines very precisely amount of calcium which enters on the alpha channels, and the whole machinery is adjusted like a Swiss clock. We want to change our focus and to understand this is not simply the highly accurate regulation of the elements, but the balance between the elements. Sometimes when the entry increases, this increased entry is compensated with an increased output of the calcium, therefore the equilibrium is maintained. Today we are going to make an attempt to understand how the calcium equilibrium is maintained in the cell, because most lecturer starts with an intriguing example, how many times our heart beats during our lifespan. So if you multiply the 70, 80 years, by the days and the course, you are going to get a huge, very big number, billions of times, and that many cycles, during that many cycles, the calcium cycling should be maintained, simplifying the equilibrium should be maintained. The crucial point in equilibrium, always the influx must be equal to the outflux. As long as this concept is maintained, we have the equilibrium. Comparing the cardiac myocytes with skeletal compared to skeletal muscles, the working volume of mitochondria, tubule, and sarcoplasmic reticulum is much larger in cardiac myocytes, showing that the cardiac myocytes keeps working during the whole lifespan of the organ, the human being. It cannot be exhausted. Soleus muscle, if it works for a while, can get fatigued or can get exhausted, but not the cardiac myocytes. The tubule size, the sarcoplasmic reticulum, everything is much larger, allowing a larger capacity for moving the calcium between the intracellular and extracellular sites. How the calcium exerts its effect. Majority of my lecture is going to focus on the direct effect of the calcium on troponin C. This is when the calcium binds to the target molecule, the protein, in all cases the troponin C, and modulates its function. This is the direct regulatory function. Apart from this direct regulatory function, in the last 5-10 minutes of my lecture, I would like to talk about the complex indirect regulatory function of the calcium. The calcium binds mediator proteins like calmodulin, kinase, and these mediator proteins are going to exert the effect of the calcium. Majority of the calcium effects, if we count the regulatory pathways, are indirect. Very few direct regulatory mechanism is known for the calcium, and the troponin C regulation is one of the best known direct regulatory function. So, if you want to maintain equilibrium, let's see what are the possible effects, what are the terms, what is the nomenclature. This rectangular object here represents the cardiac myocytes, and this is the sarcoplasmic reticulum here. The calcium which enters to the calcium, the mechanism how calcium enters is called logically calcium entry. We have the exclusion, the removal of the calcium ions. From the sarcoplasmic reticulum, we have the calcium release, and then the sarcoplasmic, when the calcium is pumped back to the sarcoplasmic reticulum, it's called sequestration. And these four processes should be in equilibrium and must maintain the homeostasis. Evidently, calcium ions do not have a mark like a red cross on their back, showing I am coming from outside, while the blue cross, the calcium ions, coming from the sarcoplasmic reticulum. There is no distinction between the ions from the different sources. Just into the fine-tuning of the processes will maintain the complete equilibrium, so that there is no direct transport for certain calcium ions in the system. Simply the transport rates are adjusted accurately. This is a list of the mechanisms I would like to discuss today. We have several calcium entry mechanisms, release mechanisms, exclusion, sequestration, and what is not shown here on this figure, we have calcium buffers in the cardiac myocytes, which calcium buffers are going to modulate the kinetic properties of the calcium movements, but do not influence the amount of the calcium. We are going to see how tune the amount of the calcium which enters into the cell via this pathway. Because of its importance, we are going to see in the later part of the lecture what happens when calcium equilibrium is penalized and it is not maintained. Many drugs were developed to modulate the calcium and used in cardiology. Today, the classification and the precise mechanism of these drugs is out of the focus of the physiology. What I would like to mention here only in our practice, in your practice in the future, the inhibition of the alpha-calcium channel, one of the most frequent intervention. The calcium entry mechanisms in the cell, usually the alpha-calcium channels do not get underactivated or say that the calcium current usually is not pathologically low. Most times the amount of calcium under pathologic condition, the amount of calcium is too much. Therefore, we have a lot of calcium channel inhibitors in the clinical practice, but we do not have one single calcium channel activator. So there is no such a pathologic condition that we want to facilitate the calcium channel. Only inhibiting is the pharmacological intervention, what we do. The calcium current flows during the cardiac action potential and depending on the cell type, which particular type of cell we have. The dynamics of the calcium current can be very different, can flow during the whole plate or in the endocardial cells in a short time, but basically has that same function to go over the contractility to activate the calcium release from the cytoplasmic reticulum. Another type of calcium channel, and this is a distinct, a different calcium channel, is the P-type calcium channel. The alpha-calcium channel name comes from the low threshold. This is the abbreviation of the low. After knowing that this type of calcium channel was named after the low threshold, we would believe the calcium channels having high threshold would be called H-type of calcium channel, but researchers are never consequent. This group of calcium channels are called transient, so this is why they have this abbreviation. We have several types of calcium channels, like N-type calcium channels. You know, those are present in nerves, for example, so neural type. Those calcium channels are not present in cardiac cells. So we have the L-type, we have the T-type, and the molecular structure and dynamics and kinematics of the two channels is completely different. So this is two distinct channels. Here is the ideal characteristic that shows L-type calcium channel activates at -40 millivolt, P-type calcium channels activates quickly, and the reason it behaves in a transient manner, because if we go back to the action potential, we can see the action potential crosses this wide voltage range during the upstroke in a short time. So within a few milliseconds, from 80 millivolt, -80 millivolt, we go to +20, +30, +40 millivolt, and that explains the name of transient, because from here we jump over here, and we can see the calcium current is very low at this voltage. So this is why it is transient, because the voltage changes too fast. The calcium channel activates, then when we reach the platelet, it inactivates. So basically, it supports the depolarization of the cardiac myocyte. It contributes to the depolarization phase, but it does not deliver significant amount of calcium to the contraction. Actually, the total fraction of calcium what T-type channels deliver are less than 5% of the total entering calcium, so it's really small. Its main role is to support the depolarization. The density, the T-channel density in ventricular cells is very low. It's practically not present in ventricular cells. We find these channels in sinus cell, excitable cells, sinus cell, AV node cell, in the thalamic, so the cells which conduct through the cell. However, when under pathologic condition, they can be expressed in higher extent in ventricular cells, and in this case, emphasizing again this pathologic condition, in this case they can add extra amount of calcium to the homeostasis and balancing the homeostasis, resulting calcium overload. Later I'm going to explain why we are afraid of calcium overload. So basically it works on the action potential but not contributes to the contractility. Two other possible pathways anti-way for calcium is the TPX sensitive calcium channel, which was discovered only 8 or 10 years ago and not present in every species. Actually, there is a big debate whether it is found or not found or human cardiac arrest. Actually, the presence of the protein was proved, but nobody could show it's a functioning protein. So it's probably it's a non-functioning channel what is expressed in cardiac myocyte. We do not know the physiological role. Actually, in the species where it is present, it's named after the TPX sensitivity. It's permeable to both sodium and calcium, but this is not the same as the phospholipid channel. So this is again a distinct protein, the calcium channel, simply sensitive to TPX, and also contributes to the depolarization of the cardiac myocytes. It's found in large quantities on the most popular experimental species, experimental animals, laboratory animals like rodents and all that. This is why it was discovered actually, and the identification on human samples is still not complete. And there is another way for the calcium to enter into the cell, and this is already proven. The presence is proven in human myocardium. The calcium channels can also use the voltage-dependent sodium channels as an entry pathway. You remember the chart, the specificity of selectivity of the channels, which means when we have the sodium channels, it's selective for sodium. However, selective is never absolute, it's never 100%. So basically, for example, in talking about human fast sodium channels, the selectivity is about 1 to 3,000, 1 to 5,000. What does that mean? Every 5,000 sodium, when 5,000 sodium ions enter into the cell, there is always one vagabond calcium ions who will use this channel and enters. Under normal condition, this is negligible. This is not a big amount. However, we should remember the sodium channels are found in big amounts on the membrane, which means the calcium ions, when it flows through the sodium channel, it also contributes to the depolarization. But as it turns out, under pathologic condition, the selectivity can decrease. For example, when the sodium channels are exposed to high concentration of catecholamines, under experimental condition, the selectivity decreases sometimes 1 to 1, which means the half of the ions which use the sodium channel is calcium. And this is a large quantity. This is a huge quantity, so under pathologic condition, this is what called slip mode conductance of the sodium channel. So this is the well-known sodium channel. It just simply allows a minor amount of calcium to enter, but in pathologic condition, this amount can be increased, also resulting in overload, therefore, as you will see later, generating arrhythmic activity. These are the anti-mechanisms. Let's see the calcium release mechanism. So these four mechanisms transporting the calcium or allow the calcium to enter from the extracellular space into the intracellular space. The next session discusses the two mechanisms, how the calcium is released from the sarcoplasmic reticulum. We know the term calcium-induced calcium, which means every calcium ion which enters through the sarcolemma activates a certain amount of calcium calcium released from the sarcoplasmic reticulum. Actually, there is an augmentation of the signal here, a so-called gain of the signal, which is high, surprisingly high. There is a species-to-species variation, of course, how many calcium ions released from the SR by one single calcium ion which enters to the cell. In human, the number is roughly 1 to 20, which means 5% of the total calcium, which increases the cytoplasmic calcium concentration, comes from the extracellular space, but 95% is coming from the sarcoplasmic reticulum. It's a relatively big gain factor if you consider this. So, one mechanism is the ryanodine receptor, and the function of the ryanodine receptor was also described in the previous lectures. I do not want to add too much information to this, only to show some photographs which was made here at our department, in spite of the warning of our first chairman, still studies the regulatory mechanism of the calcium on muscle cells. The distribution of the ryanodine receptor, this is the immunolabeling of the ryanodine receptor. We can see this very regular pattern, like a piston. The distance between two ryanodine receptors in longitudinal direction 2 micron, transversal direction 1 micron. So really like an extremely regular crystal pattern how they distribute, and roughly 50 to 100 ryanodine receptor is going to form one so-called calcium release unit. Those are the CRUs, calcium release unit, and these are located just across the dihydropyridine receptors, the L-type calcium channels in the people. This is how this calcium-induced calcium is going to be maintained, because the calcium which enters here will immediately activate the ryanodine receptors. Of course, as it was already mentioned, probably in previous lectures, the calcium released, then after activating the contractile machinery, is going to be returned to both the sarcoplasmic reticulum and to the extracellular space. The ryanodine receptor, because of its high importance, are also exposed to several regulatory mechanisms, catecholamine, cyclic AMP, PKA, PKC, amyloidine kinase. So this just simply shows many regulatory pathways converge to this channel, to this protein, in order to fine-tune the amount of calcium released from the sarcoplasmic reticulum. The second mechanism to release calcium is the IP3, the ionizable triphosphate receptors formed on the sarcoplasmic reticulum. And this is a very different story. Right? Just opposite to the very regular pattern of the ryanodine receptors. Remember the pictures like the whole cell looks like a crystal. IP3 receptors are located only two specific locations in the cell. One is the close proximity of the nuclei. So these are formed on the sarcoplasmic reticulum, but only those sections of the sarcoplasmic reticulum which are formed, which are situated in close proximity of the nuclei. And the other segment where they are formed are the two ends, the intercalated disc of the cardiac cells. So the location of these receptors, these calcium release channels, are not so regularly distributed as the ryanodine receptor. From that pattern, we can conclude easily, probably the function is not related to the contractility. It's related to gene activation and the modulation of the gene activation and inhibition. It activates the calcium which is released via these receptors or channels from the SR, enters to the nuclei, and in the nuclei it is going to modulate the DNA transcription. Someone might ask the question, how the calcium knows where it should go when it is released. It doesn't. We have so-called subcompartments, compartments in the these are going to isolate the cells. Therefore, interestingly, there is two calcium signalization pathways. However, both of them are maintained by calcium, will not mix. The IP3 signalization is much slower, while the electromagnetic coupling, rivalry receptors, work on cycle basis. So during concomitant cycle, we have the release, reuptake, we have the diastole. These regulatory mechanisms exert much longer effect. We need a tens of minutes scale to govern the metabolic activity of the cell, or the hypertrophy, or the other functions like cell division, maturation, and so on. So this is a different calcium story and not related to the contractility. However, it was shown in heart failure. The number of IP3 receptors in certain types of heart failures, the number of IP3 receptors can increase. Therefore, the amount of calcium released from the SR increases, and this increased amount of calcium might diffuse to the other parts of the cells, influencing the regulation of the contractility. But this is, as I mentioned, pathologic situation, not normal. Here I would like to show a video also recorded in our department. This is why I'm very proud of this video. It shows the calcium dynamics, how the calcium is released from rivalry receptors, and during compression, how it activates the cell. That's called Bill Gates. All of us who see this video. I'm going to dim the light. Is that the best? No more? What we are going to see here, this is a cardiac myocyte, and these sparks, these flashes here, are spontaneous calcium releases from the rivalry receptors, and the waves here, when the action potential activates the cardiac myocyte, and this propagating wave, let's say again, this propagating wave is going to activate the whole cell. So we can see these small flashes are resulted by one single CRUs, and the waves is the calcium signals, which, as I mentioned, oops, that's too much. Okay, hopefully we don't disturb those who sleep at the background. So this is how the calcium moves in the cell, and that continue. So these flashes are stars, and the big ones will called waves, and these stars and waves are generated by the rivalry receptors. And now let's see the mechanisms which are responsible for the removal of the calcium from the cytoplasmic reticulum. First of all, we mentioned is the sodium calcium exchanger, which was already discussed. This is propane, transport propane situated in the cytoplasm, which maintains a secondary active transport. This is one of the best examples for the secondary active transport. It binds 3 sodium and 1 calcium, therefore the stoichiometry for the charges of the transport is 2 to 3, so it generates a depolarizing current under normal condition when the high sodium electrochemical gradient will remove the calcium from the intracellular space. However, I would like to point out, since the transport protein is governed exclusively by the differences of the electrochemical gradient sodium calcium exchanger works in reverse direction, so it adds some calcium to the cytoplasm, but during the late part of the action potential, when we have the repolarization, this will be the main calcium removal mechanism from the cytoplasm. This is the current that is generated because of this 2 to 3 stoichiometry. This is a depolarizing current, and you can see at the beginning we have this minor feed where some calcium is added to the cytoplasm. Amount of calcium is really negligible, but this shows the ability of the protein to transfer to both directions, and the question mark is here because there is a big debate whether the amount of calcium which enters skin significantly influences the calcium concentration or not. It is shown, it is present, but the amount is minor. Most time it removes the calcium, and while it removes the calcium, it generates depolarizing current. And this is actually what gives high importance to the sodium calcium exchanger under both physiologic and pathologic conditions. Under physiologic conditions, majority of the calcium is being used by this mechanism. Let's see why we are afraid of calcium overload. Several times I pointed out malfunction of different pathways might result in calcium overload, increasing the amount of calcium. One might ask the question, isn't it good to us? We have stronger contraction. More calcium, stronger contraction. That's not the situation. When the amount of calcium increases in the heart, the sarcoplasmic reticulum, having very high calcium pump activity, is going to accumulate this calcium. This is what we actually call calcium overload. And when the sarcoplasmic reticulum is overloaded by calcium, it tends to release it spontaneously, which means the calcium release is not synchronized with the heart cycle. This is what we saw in the movie. Those flashes were calcium releases. When the package of calcium is released, this sodium calcium exchanger is going to remove it from the cytoplasm. So we might say, okay, we are ready, so the calcium which entered, the extra calcium which entered, are already transported to the extracellular space. And this is true. However, during this step, we have a depolarizing current. A depolarizing current which can be certain cases unimaginative. If this depolarizing current is too strong, too much calcium is excluded, it can generate spontaneous action potentials, and this is the source of arrhythmia. So this is why we are afraid of calcium overload, because calcium overload might result in arrhythmic activity in the heart. Sodium calcium exchanger therefore generates this very important, it's called ITI, the current that it generates, and several attempts was made in the last 20-30 years to develop drugs which can inhibit this current to prevent arrhythmic activity of the heart. By my best knowledge,
which can bind calcium, therefore can reduce the ionized calcium concentration in the cytoplasm, modulate the dynamics of the calcium, but they do not contribute to the determination of the absolute amount of calcium. They can shift the timing of the peak, and they can facilitate the sink of the calcium during the diastole, but basically they do not act or remove calcium from the system. One of the most important buffers is the calcium, the protein, which is found in the cytoplasm reticulum. This is the photomarker. Here we see the peculius and these dense bodies here are the calcium, and this is the model representing the cytoplasmic reticulum. Calcium binds the calcium with high specificity. The stoichiometry is extremely high, roughly 40 calcium ion bonds to every calcium molecule, so it's a pretty much calcium molecule, but it also explains why the free calcium concentration in the cytoplasmic reticulum is not high. So the cytoplasmic reticulum can store large quantities of calcium without having extremely high concentration, because the majority of the calcium is bound to this protein. The dynamics of this protein is very exciting. In calcium-free form, the protein, the fragment of this protein, and the bigger is the number of calcium ions, what is called, the protein takes a more globular form. Finally, it is going to form superstructures, which interestingly are located in a close proximity to the inner aperture of the ion receptor. Therefore, this very small arrangement allows the very rapid release of calcium, because when the ion receptor opens and the calcium starts to flow out, this buffer are close to the release site and can provide further amount of calcium to support the conductivity. So this is the exact mechanism how it accumulates is not known, but the localization, the co-localization, the granular receptor is already good. Mitochondria. Mitochondria also function as a calcium buffer, as the data shows, roughly 30%, one-third of the calcium, what is released from the cytoplasmic reticulum, sinks in the mitochondria, enters to the mitochondria during the early phase of the diastole. Of course, mitochondria will not store it for a long time. During the late diastole, when the cytosolic calcium concentration is reduced, it is going to diffuse back slowly to the cytoplasm, and the same circle will return the calcium to the SR, which are responsible, which certainly responsible for the early level. So practically this is a temporary storage place for the calcium. During the diastole, it facilitates the decrease of the calcium concentration in the cytoplasm. However, if we remember that in mitochondria we have high concentration of metabolic enzymes, and those metabolic enzymes are also calcium sensitive. Actually, calcium activates those metabolic enzymes. We can see the small arrangement because the same calcium signal, which is responsible for the conductivity, also adjusts the production rate of the ATP. So the same calcium pathway will serve two signalization functions. One is the conductivity, and at the same time it adjusts the ATP production rate to the need of the pumping activity of the heart. When we have the pumping activity, more calcium is released, therefore the enzymes are activated. These were the direct regulatory mechanisms of the calcium, and as I promised, only a few words I would like to mention two indirect regulatory mechanisms. One is the calorimetry. This calorimetry we will also discuss in detail potassium channel is shown here, and the intracellular side is activated by the carnitine. Another indirect way of activation is the carnitine kinase. Carnitine kinase is an amazon protein, kinase protein in the cytosol, and the carnitine calcium carnitine complex binds to the carnitine kinase that activates it. After activation, 8 to 12 carnitine kinase is going to form superstructure, and it stays inactivated from and there, and this kinase is going to phosphorylate several cardiac enzymes. So this is not only secondary, this is a tertiary activation of calcium activates, carnitine activates, carnitine kinase and carnitine kinase activates, as this pathway shows, several calcium channels and contractile mechanisms in the heart. The carnitine kinase are also modulated by catecholaminergic regulation and other. So this is a big nodal point of the regulation, but the most important regulator here is the calcium carnitine system. I mentioned with other side CRP. Under pathology condition, when it is overactivated, it will generate pathologic action potentials and morphological disorders in the heart. And finally, summarizing all the regulatory functions of the calcium that we discussed, usually I use this map to scale my students at them. We discussed the calcium cycling, the electromechanical coupling. We discussed the IP3 mechanism, which acts via gene modulation, and we mentioned the calcium has also an important role to regulate the morphological action potential. Therefore, it also modulates the rhythm generator of the heart. And what happens when things malfunction? What happens when the calcium homeostasis is not maintained, when the calcium equilibrium is not maintained? Ladies and gentlemen, the main force of defective calcium handling results either contractile failure, reduced contractile activity, or incomplete relaxation. Surprisingly, this is the most frequent form of the heart failure. Then the relaxation is not completed, or the pathologic excitability results in arrhythmia, and through the IP3 gene activation pathway, long-term imbalance in equilibrium results in hyperactivity, morphological disease. These are all resulting by pathologic calcium homeostasis. And the significance